Hi, my name is Jabran Zahid. I'm a researcher at Microsoft Research working in the immunomics group, and today I'm going to be telling you about how we are working to learning how to read the adaptive immune system. This project is a deep collaboration between Adaptive Biotechnologies and Microsoft, in which we're trying to bring the science of immunology together with the power of the computational power of artificial intelligence in order to build better care for healthcare using what we call immunosequencing. So we're learning to decode the immune system to diagnose diseases. And this really requires several steps and a deep expertise coming from the two organizations. So the first step of this is we need to draw blood samples from individuals. And so we take a vial of blood from donors and we do what's called immunosequencing. This is really the expertise of Adaptive Biotechnologies. They take hundreds of thousands to millions of T cells and sequence genetically the T cell receptor. And on our side, in terms of the data, we get genetic sequences of the T cell receptor. Using these data, we apply sophisticated machine learning techniques in order to try to learn how to decode the immune system using these genetic sequences. And the techniques that we develop and the diagnostics we're developing hopefully lead to empowerment of healthcare for humans. So here's the expertise stack that we're working with, in which there's a deep expertise in terms of the immunology coming from Adaptive. So we have an immunologist and clinicians who are deeply involved in getting the data and in terms of sequencing it, and that feeds into the computational biologists who begin the analysis. And then on the Microsoft side, we have the AI researchers who are working to understand these massive data sets. And there's a back and forth. As you can see, the data flows down from Adaptive, algorithms and models flow up from Microsoft, and it's actually quite a synergistic process of building out this diagnostic platform. And so to start with, I'd like to discuss the immunology and how it all works. So how do T cells actually learn to learn about pathogens? So the immunology starts with a bunch of T cells. And then there's the human cells. And those cells themselves have viruses or can be infected by viruses. So there's genetic material inside the cell here. And so that genetic material gets broken up into little pieces. It gets chopped up inside a cell. And that then gets transported to the surface of the cell via what's known as these molecules known as HLAs. So if something like a virus comes in, the virus itself will get chopped up. Here's the COVID-19 virus, for example, and then it will get transported to the surface. Now, if there's a T cell in this bunch of T cells that recognizes this little piece of the virus, it will initiate an immune response and it'll clonally expand. And this is really the critical ingredient in our ability to build diagnostics based on the T cell immunology. So the specificity is determined by the T cell receptor. So this TCR bit is what we actually sequence at what Adaptive Biotechnologies sequences. And that completely determines what peptide or what part of the virus, what antigen a T cell will respond to. That signal is clonally amplified because of clonal expansion, and that's the signal that we try to read out. So for a given disease, you can have tens of thousands of these T cells responding to hundreds of antigens. So the virus gets broken up, creates all these antigens, and various T cells respond to those antigens. To give you a sense of the challenge of the problem, let's take a look at the numbers. So it's currently believed that there's something on the order of 10 to the 15 possible T cells. These T cells are produced via a quasi-random process. And of those T cells, any given individual, any human will have on the order of billions in their body just floating around. Not all of them will be unique because of this clonal expansion, so on the order of millions of unique TCRs. Of those, we sample using the immunosequencing, we immunosequence on the order of 100,000 to a million of these billions of T cells, and we refer to this as the repertoire. Of those, for any given disease like COVID, we anticipate on the order of 1,000 to 10,000 T cells are specific to actually COVID, are responding to that virus. Other T cells may have responded to other illnesses in the past. Some may not have responded to any illness at all thus far. And then of those, the signal we really leverage is the shared signal. the signal that we can see, the T cells we see amongst many, many different individuals who all have the same disease. So this is really a new approach to diagnostics. Given the T cell repertoire, can we predict disease? And the answer is absolutely. And we've done that with COVID. So we are able to, using this technology, detect a past COVID infection. So here is our basic signal. The way the setup works is we have a case control setup. We have, let's say, on the order of 1,000 individuals that we know have COVID and 1,000 individuals in our control sample that we don't know do not have COVID. And we just ask the question, are there more individuals, are there T cells in these individuals that have COVID that occur in a statistically higher frequency than the individuals who don't? And those T cells are the ones that we anticipate are responding to the virus. And so for COVID, we have something like 10,000 T cells we think are responding to the COVID virus. And the diagnostic is essentially asking the question, how many of these 10,000 T cells does any one individual have? And so what this plot shows is on our sample of holdout patients, people who weren't involved in deriving the diagnostic, the signal. So this axis, the y-axis, shows the number of sequences each of these individuals has. And the x-axis here shows the depth of the sequencing, because the sequencing depth does vary for various reasons. But the upshot of all of this is that the orange points are people who have COVID, and the blue points are people who don't. So people who have COVID have elevated counts of these sequences. And the most important, one of the most interesting aspects of this is we have many, many cohorts spanning many continents and many different countries, many different racial groups. And regardless of where they come from, we see that we're able to diagnose and discriminate individuals who have COVID from people who don't. So what this means is we have the ability potentially to build universal diagnostics for detecting diseases. Here, this plot shows, or this table shows, that the sensitivity of our diagnostic increases as a day since the infection. So, our highest, it takes a while to mount the infection, but then once we get to something like 2-3 weeks in, the diagnostic out to several months, or the signal out to several months is quite good. This is the sensitivity of our ability to tell whether someone has the disease or not. And so what this really says is we can use this technology to understand past infections of diseases. And so as we build out this platform and start detecting more and more diseases, we hope that we will be able to read the immune history of individuals. So one of the key aspects of all of this is we don't actually know, given the way we do our diagnostics, where we look at case control setup, whether the actual T cell is responding to something in the virus. But we have a separate set of experiments that allow us to begin to understand that question. In these sets of experiments, we take a bunch of the virus, chop it up into pieces as we think the body would and in the presentation step within a cell, and then we throw a bunch of T cells at it and ask which T cells are sticking. And what this plot shows on the x-axis is the genome of COVID. And then the orange, for example, the surface glycoprotein is the spike protein region of that genome. And these blue bars are basically showing where we have T cells hitting the virus. So these are candidate T cells that are responding to the actual antigens. And now we know the antigens because we've chosen the antigens that we presented. And what's exciting is that using this technique combined with the case control setup that I described, we are now able to convincingly demonstrate that we are actually hitting the antigens that we think we are, because we can separate out and identify which TCRs are hitting which antigens. And here is a really interesting example of demonstrating the validity of this. So what's being shown here is, this is again our signal versus our signal plot as I showed before, but this also has vaccine recipients on it. So these people in red are people who've received the vaccine. Now for COVID, the vaccines only target the spike protein. And as a consequence, we would only expect an immune response targeting spike. And so when we separate out the T cells that are specific to spike, this is what it looks like. And indeed, people with vaccines separate from the blue controls. If we look at the rest of the response, which is not spike, we see that while the people who have a natural infection do separate out from the controls, the individuals who've received the vaccine no longer do. And so what this is telling us is, to some degree of resolution, we are able to discriminate between individuals who've had a natural infection and vaccine, and therefore confirms that we are able to identify T cells and the antigens that they're responding to. Ultimately, the antigen map will be, we hope, a platform whereby we can take a T cell repertoire, not even know the antigens, infer the antigens, because given enough examples, we will learn, given a T cell, what antigen it responds to, and then eventually use that platform to predict many, many diseases, including cancers, autoimmune disorders, and various infectious diseases. Thank you very much.